Hello and welcome, everybody. I hope everyone's happy and well. Welcome to week 9 lab session. So we're on the home straight. This is actually going to be our last live cast lab walkthrough for the course. So welcome and thanks for joining us. So this week we're going to delve right into some RNA-seq analysis. We're going to jump up in complexity a little bit and start analyzing some data from this manuscript that actually appeared quite a few years ago in PLOS One. This is from Himes et al., and I discussed it a little bit in our pre-course, or not our pre-course, in our pre-lab video sessions for this week. We just had a couple of them. So the idea here is they were analyzing a set of glucocorticoid steroids in their work. These are commonly used for treating inflammation as associated with asthma, for example. And so these authors, what they did was to try and dig into the mechanism of action of these compounds to try and come up with better therapies ultimately in the future and understand why some patients aren't as amenable to treatment via these types of steroid kind of inhaler-like treatments as some others. They wanted to understand what was going on when you treated different cell lines with one class of these glucocorticoid steroids called dexamethasone, one of these common steroids that are used. So they started off to do RNA-seq analysis. They looked across a couple of different airway smooth muscle cell lines, or ASM lines, and we discussed this a little bit in our previous video, so I'm not going to dwell on it too much here, but just to set the scene so we know the kind of data that we're analyzing. They went and they used top hat and cufflinks. These are the same tools we used in last week's lab session, and they found many genes that were differentially expressed. And then they focused in on one in particular that they had studied previously and that was known to be associated with lung development. There were actually variants of this gene from GWAS studies that were associated with resistance to these type of bronchodilator responses that we're after to try and stop this swelling and inflammation of the airway. So if there are certain variants in certain patients, they wouldn't have this positive response that we would be looking for with these drugs. So it was well established that this drug or this drug and the kind of asthma phenotype indeed, and its treatment with this drug is affected by variants of this gene. So it was a good one to go after, and they verified that indeed expression levels are regulated when you treat them with this drug. So we're going to go and dig into this data a little bit more because there's actually lots of really nice findings that we could get out of this data set, and we're going to see what we can find out. So you see the kind of how to analyze this data with a view of being transferable across different data sets that we could go and download and analyze in this way and mine for interest in biology that we want to investigate further. Okay, so we're going to start from where we kind of left off last week. We're going to take the output of that top hat and cufflinks work. This is this count matrix. Again, this has genes in the rows and samples in the columns, so it looks a little bit like this. This is what we call our count data matrix in the terminology of the tools that we're going to use today. This is the DSeq2 package that we're going to install and use to analyze this data. But it has, if you see, I've just labeled them gene A, gene B, gene C, gene D, gene E going down the rows here. This would be the real genes in this data set. And then we have these different cell lines that these authors were investigating in this case. So they had 4 different cell lines, and they're going to treat each one of those with drug or with a control to see the effect. Now, there's another thing that we need to analyze this data properly. That's the metadata about this count data matrix. It's like which column in it is actually drug treated, which is this cell line, which is that cell line, this kind of thing. So the call data, or this metadata here, it'll describe the setup of your experiment, how we want to actually make our comparisons. Do we want to compare drug treated versus control? Do we want to compare this cell line versus that cell line? These sorts of things. We're going to need to know which columns that data is in. And this all comes from this metadata, the second little table that I'm drawing here called call data. That's going to tell us how we're set up and how we're going to do these comparisons. So we're going to go between these two tables to figure out what's going on. Okay, and the key point, as we mentioned in our video for DSeq2 and indeed for the work that we're going to do today when we dig into it and do it kind of manually first, is we need to have a good correspondence between those that call data, that second little table here, those rows, they have to match the columns and the count data. If they don't match, you're going to be comparing the wrong things, right? You're going to be taking the control as treated or the different cell line as this cell line. It's just going to be a mess. So the key point is that we have a match between these two little tables so we can do our bookkeeping and be comparing the right things against the right other things. Okay, so let's get started. Let's get DSeq set up. And hopefully you had a chance to do this already. I'm going to do it too. I'm going to go and install this as well here on my RStudio session just to make sure. So the first thing, as you can see in the text box here beside my head over here, is we're going to use bioconductor package. We're going to use this bioconductor package called DSeq2, and we should have bioconductor all set up, but just to make sure, we're going to run that first. Add command here. This is going to go and get a package from CRAN called BioC-Manager, and that does the job of looking after all of the installation of bioconductor. If you remember back to that class, that section where we had an introduction to CRAN and bioconductor packages, I had this image of an orchestra, right, that we're all playing together, this idea that the bioconductor package has lots and lots of packages and they all play nicely together to make music, and that we all reuse parts of different packages, data formats and imports and outputs and these kind of things, and share them between different packages is really key in bioconductor. But it means that often when we go off to install one package, like we just want to use DSeq2 here, we're actually going to have to install quite a bit of the orchestra to make this thing play the music we want, because we're sharing all this infrastructure across these packages. And that's that conductor slide, if you remember the orchestra slide. So let's go and see what this looks like. So we're all on the same page. I'm going to pop off to RStudio here. I have it open here on the screen behind me. So if I've got a blank RStudio here, I'm going to start, as always, by opening a new RStudio project for this class, and then I'll do an R Markdown document as well that we'll use to store our work and upload to Gradescope later on. But let me start a new project up here. So I'm going to go up to the top of the file, new project. It's going to bring up my now familiar set of click-through options. I'm going to say new project. I'm going to make sure I'm working in the place where I'm working for all this class. So that's off my desktop, and I've got a little folder for this course. And I'm going to call this week 09. Okay, and I'm going to say create project. And that's going to flash. R will restart itself, RStudio, reset, and I get my R prompt, and everything's right with the world. I've got this clean, empty environment. You see in the panel right at the top above my head, it says empty. The environment is empty. I've got no objects or no detritus in here from previous sessions or anything like that going on here. So that's where I want to be. And now, before I even open my R Markdown document, I'm going to go and install this package like we had on the slide. I'm not, I sound like a broken record because it's important. I don't install packages and write the code in my R Markdown document. I do it here in my console, in this panel, right? The R brain over here. So I'm going to go and do this. Just make sure that I've got it. So I'm going to do install .packages, and RStudio is trying to help me here. Let me make this font a little bigger. So I'll click the little broom here to clean up so we're up at the top so you can see what I'm typing more easily. So I'm doing install .packages, and I'm going to do the BioC-Manager. BioC. Oh, and I have to type a B. BioC. So this is going to go and install this package, or look for it if it exists. If I make a typo in it, if something like that, it'll say package not found, right? If it's a name of a package that doesn't exist, it'll say, bleh, right? Can't help you, boss, right? You have to spell accurately and do all those things accurately, as we've discovered, much to our annoyance and frustration, right? Normally, it's just typing accuracy that kills us here and causes these error messages. So now I've got the BioC-Manager package installed on my local computer here that I'm working on. And I could, we've seen before when we're using packages like Bath3D or ggplot2 or dplyr or any of these other packages, you would type library, the name of the package, and then we can just go and use all the functions from that package. And we could do the same here. We could type library BIOC manager and then use it. There's actually only just one function I want to use from this package. So I don't want to load the whole package. I'm just going to call that one function from this package. And to do that, I'm just going to type the name of the package. So BIOC manager. And you see RStudio is helping me. It's giving me this prompt. Do you mean this one, boss? Do you, right? Like the little puppy dog wagging its tail. This one, right? So it's BIOC manager here. And I'm going to say, yeah, that is what I mean. You see it's got the 2 colons after it. That means go to the package and then pull out a function from this package. And the one I want is the install, the second option here, BIOC install. And if I just run this without any argument in it, so nothing inside the regular brackets or parentheses here, it's just going to set up the base bioconductor. It's going to bring along those core bioconductor packages. So I'm going to run that first, and then I'll do dseek2 afterwards. You can always tell it to install an individual package. So here it's asking me something. This is one of the reasons, one of the many reasons we don't do this in our R Markdown document or put this code in our R Markdown document, because it's asking me a question. It's saying, look, boss, there's some packages on your computer that are that new versions are available for. Do you want to update all, some, or none? And I'm going to say none in this case. You might have a long list of packages here if you haven't kept your system up to date, for example, or if you haven't installed Bioconductor before. For example, there might be a long list of them, but we're going to start, at least initially here, by saying no to this. I'm going to say N for no. And that's good. Happy days. Okay, so I am then going to recall the last command. So I'm pushing, here are my keys, I'm pushing my up arrow key to recall my previous command. I'm not typing it all out again because the command I want to right now is install, but I'm going to put D, E, and it's capital D, capital E, capital S, E, Q, 2. dseek2. So there's an uppercase D, uppercase E, uppercase S, and then a small case EU, and then the number 2, and I'm putting that inside quotes. This is the package I want to install, and I'm going to say, go off, please, and install this dseek2 package. It's going off and getting it, and it's telling me, oh, look, there's some other packages here, boss, that it wants to upload. Now, there's only a few of them here. I could say A for all and go off and do them. I'm actually going to say no here as well. And I'm good. Now I'm going to check, because I just say no kind of approach, I'm going to go off and check here if things are okay with the world. I'm going to go and check if the dseek2 package actually, I can load it and use it. So I'm going to type library, and I'm going to do deseek. So it should be one of these options here. It's dseek2 is the one. Make sure you have the 2 there. So now when I push return here, it's going to make the package available and all the functions within the package available for me to do my work, right? But it will also print some messages about bringing along the orchestra, loading up all the other packages that dseek2 needs to work, right? Because it's this bioconductor package and it's going to use all these different methods from these different other bioconductor packages. So don't be alarmed that a lot of text goes past, but let's just spend a minute seeing what that text is, okay? If you see a big message that says error, the word error, then we got to pay attention. If it just says warning, we'll check what those warnings are, but we should be good to go. But if it's error, then we're going to have to go back, and where we said no to those questions, the solution usually is to say, yes, let's update those packages, okay? So I'm going to say load it up, and lots of red text. It's red font because that's the theme that I have. Doesn't mean it's bad text, right? So let's go and have a look at what's being printed out here. So lots of stuff flew past here. This is the orchestra coming along, right, for the ride here. So here's where I actually issued my, oh, sorry, I've highlighted too much, but this is the line that I just typed. And then these are the messages being printed out. It says here, loading this package, loading this package, loading this package, loading this package. Oh, and this package is kind of this biogenerics package, and then these are functions here from this package that have names similar to functions in other packages that you had loaded. So I'm masking those. So now you use these functions, right? So there's a whole lot of stuff going on here, like the interquartile range, this function called mad that doesn't refer to me. That's a function within this package, stats. So there's a normal package called stats, but when we load this biogenerics, there's a new version of mad that's loaded over the top of it. So we're going to be using that one. Now, all these other things are masked. And then it's loading I ranges and genomic ranges and genome info, DB, database, summarized experiments, biobase. This is our orchestra, right? It's like the one up here coming along. This is like the orchestra, the player coming in with her coming in with her big double bass and bumping all the other things out of the way and saying, right, here, I'm here. Let's bring the percussion in, all these other packages. Okay, I'm going to stop with the acronym and the metaphors here, and let's get started. So there's warning messages here. It says DSeq2 was built under a previous version of R. I installed from binary version, not from source. If it asks you, there's a later version. Sometimes this may ask you, oh, there's a later version of this package available, but it's not available in binary format. That means like a compiled version. I'm on a Macintosh computer here, or a Mac, and you can install. You don't have to install the package from source code. You can install it from these pre-built binaries. That's what I did. But it might say there's a later, greater version, but the binary isn't available yet. Do you want to install from source? I'm going to say no, because sometimes we don't have the compilers for that available on student computers where we are. But I'm installing the binary versions. They were built under a slightly earlier version of R. I'm on a later version. But it's just warning us about that. But we're good to go here. There's no error messages. There's no big problems with the world here. Now, if you do have an error message or something confusing that you can't solve by answering yes to those questions, to the install issues, I want you to go to our class website, please, and check out the guidelines. There's more detailed guidelines. So I'm going to pull these up here too. Just bear with me for a minute. I'm going to go to our class, go to our website. That's so actually we're on the schedule. Here we're on week 9. So this detailed bioconductor setup instructions. So if you click here, it'll open a page where it basically has the code that I typed in here and some notes about the install process, and if things went wrong, there's some guidance about where to go for help. If you still can't fix it with this, post on our forum, please. Post on our Piazza forum, and I'll have a little look there too, along with the IAs, and we'll be able to get you up and running. Okay, so it might, as always, because we're doing this, bringing the whole orchestra along, there might be some confusion, or it mightn't work first time. There might be some little workarounds that we need to do. So just let us know, please, and we'll get you up and running. We're going to need this DSeq2 package a little bit later on in our lab session for today. Okay. So let's go and actually do the work then, right? So here we have our class website for today, or our class section for today, and we should have this one just below it here saying the hands-on section worksheet. This is a HTML. I actually have it open here already, but I'll just open it again. And I'll close this older one. So here's our instructions for today. You can see the same image that I had in my slides. Here's the airway smooth muscle cells in this normal airway. Here's the inflamed airway, where we've got a much thicker airway wall and mucus in it. This kind of conditions that we want to ameliorate or help treat with these kinds of compounds that we're looking at today, these drugs that we're looking at. And then I've got a lot of verbiage about the study. You can link off to where I got the data from. This geo database is where I just went and got the the data for today's lab session from that study. Here's our bioconductor and DSeq2 setup, and we're all good. So I'm going to then this also notes there's some references here that are recommended reading as well. These are talk about the overall differential expression analysis kind of workflows, RNA-seq analysis. There's some nice papers here that, if you're so motivated, will give you more information and more background and different approaches to do this. We're taking one approach, but as always, there's many different workflows and different tools to use to do these sorts of things. Which ones you use often reflect your personal biases and what you have grown up with in some respects, or what you've been taught. But here I'm following on from what the authors of this manuscript did. We're doing it this way, right? So that's the idea. And here's the same image again that talks about the call data and the count data. The count data being the main thing, right, with our genes in it and our experiments in it. So let's go and read this data into R. Let's get on our Markdown document and let's read this data in. So I'm going to scroll down here and see section 2, import your count data. And actually, I see there's links to these two files. They're CSV files. So I'm going to put these into my project directory. I'm going to click on this here and say, so I'm clicking with the other mouse button, or control clicking on a Mac, or the other mouse button on your PC, and where it says, so if I just clicked on it alone, it would just download to my downloads folder, but I want to download it into my project directory where I'm working. So I'm going to say download linked file as, and here I'm going to go to my desktop, courses, BIMM-143, and I'm going to find week 9. So I've just navigated to the location of my work, my RStudio project that I opened a moment ago, and save it in there. Off it goes. I'm going to do the same thing for the next file. So I'm going to download linked file as. It should have the same name. If you can see up the top there, it's maybe a little small. I'm putting this inside my week 9 folder, and I'm going to say save. So then, if I go back to RStudio, do that here, flip over here, and excuse me for 1 second. Okay, so here I have all that verbiage from DSeq2. I haven't opened my R Markdown yet, but I see above my head here in this panel right behind me the files. I see the airway underscore scale counts and the airway underscore metadata. So that's the two things that we need. Remember, our call data and our count data. Count data is the first one, the call data, the metadata is the second one, and I'm going to read those in. But before I do that, I'm going to open an R Markdown document as always and save my work and whatnot. So I'm going to go and do that. I'm going to do File, New File, R Markdown. Oops, excuse me. So here's the window. I'll make mine PDF because that's what we're going to use to submit to the Gradescope. The title, let's go and I'm going to get that from the I'll just call it week 9 in fact. Call it lab week 09. The author is me. That should have your name. I'm going to put my PID number in here as well, my imaginary student number, UCSD number, PID 911, and I'm good to go. So I just open it up. This will have the boilerplate text, and I'm actually just going to delete all this stuff. So I just have my header up here. It's got PDF document in it. It's got today's date in it. I'm going to put a line, a level 1 header here, and I'll just copy and paste transcriptomics. That I copied this from the lab sheet, right? Transcriptomics and analysis for RNA-seq. I'll write some notes for myself. We're going to data from Times et al. I could paste in the reference from our from the class website about the work that I'm doing, right? So keep notes for yourself that will make sense to your future self when you read them, or someone else when they come to them at a later point. So I'm just going to go and read this data in. So I'm going to use the read.csv function because it's CSV, and I'm going to read in that Irway scale counts first. Irway, I'm going to put this in quotes, and then use the tab button to help me. So the tab, once I push tab, it'll give me the two options. Let's read the scale counts first. Now I'm going to set this option, should be the default, but I'm going to make sure the default in the new version strings as factors equals false. This is just, then I'm also going to set the row names equals 1. And I'm going to make sure that I call this thing the same output. I'm going to make sure I call it the same object that I assign it to as I was working on in the on the website, so we don't confuse ourselves. So here's the class website. I'm going to say import count data, call data, and I call it counts and metadata. So you could, of course, just copy and paste this. I'm writing it out because it's good for my soul to do this. So counts, and maybe I'll cheat and copy this one. So I'm going to go back to R here. There we are. So this one was called, it's called counts. I'm just going to try this one first and see how things work. So it should find it. It's in my project directory. This file. If the file isn't there, or you type the name wrong, it's going to say can't find file, or error connection, can't connect to file. So that's a typical message. Like, for example, if I had misspelled this, so I'm just deleting some characters from this name and try it again, there's going to be a message coming saying error, cannot open the connection, right? Because I've mistyped the name of the file, right? I've deleted some letters from it. So that's a common error message that you've seen before, I'm sure, because it's trying to read a file that either doesn't exist, as in this case. I'm going to make sure I type it right. Just going to use the tab button to autocomplete. The tab key satisfies my inner laziness, but it also makes sure I type correctly, right? So it serves two purposes, right? And I like that. Make sure that I don't make a silly typo. So that's my counts. Let's have a little look at it, right? So this, I can see up here in the very top environment panel window that there's actually a lot of stuff in it. There's over 38,500, 38,694 rows in this thing. That's going to be genes. Let's have a look. So I'm pulling it up here. And it's got, because I set the row names equals 1, it's got this gray first row names column that has these ensemble gene identifiers, right? So they don't mean much to me at the minute because they're ensemble identifiers. They're not, HBB or kinesin or cytochromes P450 or something, right? Some names that I would know something about. They're ensemble identifiers for the ensemble database. That's the way these authors worked. And then also in the columns, I've got the names of the actual samples that we got, and they're called SRR103, right? What's that? Is that drug treated or control? I don't know, right? Yet, I don't know. And then there's another one, right? So it tells me there's 8 total columns, okay? And there's 38,694 rows. Let's check that in our Markdown document. What I'm going to have to do here is I'm going to have to open up and read the metadata as well to make sure I know which one of these columns is the drug treated and which one is that cell line, which one's the control, etc. Okay, that comes along in that metadata file, the file that I haven't read yet. So let me read the metadata file in to check how things look, and then we'll have a little quick summary and see how many genes are in this data set. Hint, it's the number of rows, right? There's 38,694 gene entries, rows in this data set. So it's a reasonably big data set. I'm going to go back to my R Markdown document. I notice as I'm clicking on it that it's called on title 1. It's got a little star, it's in this blue font. That means I haven't saved it yet. So probably a good time to click the little save icon. So I click that. And it'll ask me, do you want to save it? And I say, yes, please. Because I don't want my computer to run out of battery or something else to go wrong and then I lose my work. So I just click save. And it should appear in my files panel right above my head. See, it says now week 9, the RMD extension document. So I've got my file saved. Let's go and read the, that's their message from before. Let's go and read the other file. I'll do it here too, in the same chunk. So I'll put a little comment there, read count data and all data files. So I'm going to click play, make this bigger. So it's a little wrapped because I'm in such large font here. I'll actually just make this a bit bigger here. So you can see it easier. So here we go. We've got our metadata and our count data here. Let me have a little look at the metadata before we go any further. See what that looks like. So here it is. It's got a column called ID, a column called DX. Remember, our drug was actually dexamethasone here. So this is the name of the shorthand, the short name for this drug, Dex. And it's telling me that this first ID was a control. The second one here was a treated. Okay, where I am here, and then control, treated, control, treated, control, treated, as we go down this metadata file. Or if you notice the ID column SRR103905, if we went back to our counts, which I still have open, that should be the first column here. See, it ends in 508, and then 509, 512, 513. Let's remember that. 508, 509, 512. Let's have a look. 508, 509, 512, 51. Right. So visually, things are looking okay. Not mad or crazy. And it looks like it's paired control and then treated for this particular cell type and 6111. And then we've got a different cell type, control and treated. Different cell type, control and treated. So there's 4 different cell types and a control and treated for each, right, where we treat with this Dex drug or not. So that's the idea. So now I kind of know what is in my count data. I know that the first column is control and the second column is treated, and the third column is control and the fourth column is treated. Get the idea? Okay. So let's go back here. Actually, I have if we go to our web page for this class, we'll see what the questions are here. So it says have a look at them. We use the view function or click on them to get the head data. So it says how many genes are in this data set? Well, let's answer that. Question 1, on how many control cell lines do we have? Let's take these 2 questions and answer them. Notice that there's the little I have a hint, or I need a hint. Not that I have a hint. I don't have a hint. But you can click on this optionally to get some extra information. Let's try it without picking the hint for here, and let's try and answer these questions. So I'm going to go into that. So I'll put these little chevrons in front, or these little more than symbols, because when I render it, and it's going to appear in a different font. Oh, let me go back to, I didn't click on our studio. Apologize. Let me do that again. So I've just pasted in those 2 questions here that you can see them in the green font here. So how many genes are in this experiment? Let's answer that. So how would we get this answer? Well, we could just do n row of the counts. That would tell me how many's in it. Let's run that. It tells me there's 38,694. I could write it if I was so motivated. I could write a sentence here, say there, like we're in elementary school, right? Answering one of our teacher's questions, right? There are, now I'm going to use this little back tick, there are n row counts close the little backticks. Genes in this. So when I do this, so it doesn't tell me in our markdown document what the answer is. When I knit this document, so I'm going to save it and I'm going to knit, it's going to insert the answer where it says that, where it has the little R, it's going to, our markdown is going to know that it has, so it's already done, but I haven't showed you it yet. So here, so let's have a look. So here's my my PDF, and when we see it here, it says there are 38,964 genes in this dataset. Okay, question 1, how many genes are in this dataset? There are, so it's inserted this answer, our markdown answer here, into the knitted document. So hopefully you can see that there, how that works. So that's one way to do it. Of course, you're welcome just to put in the code chunk and answer it directly, like the way we just did here. So how many control cell lines are there? Well, there's going to be 4. That's what we could see by our eyes. But let's see, how would we? I'm inserting a new code chunk. I'm using the shortcut key to do that. You can always use the insert button and click R. I'm using the command option I buttons on my Mac, or control, the little window key I for insert on a Windows computer to insert this. So how many control lines are we? Where do we look for that? Well, we have to look in that metadata file. Let's have a look. So there's the metadata, and I'm going to take the column dex. So here I'm using the dollar syntax to get to the different columns in this, and our studio's been very helpful. It tells me there's an ID column, there's a dex column, there's a cell type column, there's a 4th column called GeoID. I want this dex one because if I print that out, it tells me control treated, control treated, control treated. Okay, and I could just do it. There's a number of ways I could do that. I could just say table of this, right? That would tell me how many of each there are. So there's 4 controlled and 4 treated, for example. It's one way to answer that. Or you could say equals equals treated, right? Another way you could do this, if we're going to be a little bit more, apologies, there's a plane helicopter going over at the minute, if you can pick that up on the sound. So this was our column here. Let's have the control treated. If I wanted to know how many were control, or which ones equal control, I could say equals equals. Okay, so the equals equals is going to say, does the thing on the left of the equals equals match the thing on the right of the equals equals? And if it does, it's going to say true, and if it doesn't, it's going to say false. I could do control like this, and it's going to tell me, let's predict before we run it what it's going to do. It's going to say false, or sorry, it's going to say true, and it's going to say false for the next one. It's say false, true, false, etc. Right? Let's check. So it says true, false, true, false, true, false, true, false. So how would I get the number from this? Well, remember that Boolean trues and falses, they can be treated as numbers if we do math with them, because trues are 1s and falses are 0s. So one way we could do this is we could just take the sum of this. Sum. And it's going to count up. It's going to be in the vector here, it's going to be 1 plus 0 plus 1 plus 0 plus 1 plus 0 plus 1 plus 0. So it's going to be 4 1s, and it's going to be, the answer is going to be 4. So there's 4 cell lines. Okay, that's another way to do it. You know, the table method, if you want to just get a summary of what's in a vector or whatever, that works fine too. But this equals equals is, I wanted to show you because we're going to use that in a little minute. Right? So I'm going to save my work. Command S here just to save my document. And I'm going to look at what's next in the workbook. But what it's going to likely be is we're going to have to go and get the values for the counts from for the control and the values for the treated and do something with them, because we want to compare, does this drug do anything, right? Is this drug affecting this data set. So to do that, we're going to have to go and find the answers for the control, and by answers I mean the counts for those control samples and the counts for those treated samples. So let's pop back and make sure I'm not delving too far from the script, from what's been asked. And we'll pop over here. So we've answered those two questions. We're in section 3, this toy differential expression. I'm going to copy this actually, this text, because I'm going to paste it in. So the note here, it's in bold. It says, the analysis that we're about to do is for demonstration purposes. Don't actually do it this way and do real analysis this way. What we're going to do is kind of code up the inner guts of the dseek function that we're going to use, just so we know how it works. Right? We're going to actually go and compare these, as you can see in this table that we've just pulled up on the web page. I've colored them up blue and gray to try and emphasize this paired nature. It's control and treated, control, treated, control, treated. We're going to have to go and find those control experiments, get the level of gene expression for every gene in those, compare that level of gene expression to those where we added the drug, and then see which genes are different when you have the drug. Okay, so we're going to do this kind of manually. We'll see that in section 4, where we actually do dseek2 analysis, there's a robust way to do this, and we'll talk about why it's robust. But before we just run that, and then you wouldn't appreciate what's going on, we're going to actually do it manually ourselves. So the idea here is, as we're talking about in the class website, here is that note that the control samples of these names SRR, right, these really catchy column names, so the one that ends in 508 and the one that ends in 512. So that's telling me this one is control. Oh, sorry, I didn't need to highlight them all. It's tricky to use the mouse in this mode. So this is control, and this is control, and this is control, and this is control. So I want to go and access, essentially what I want to get is get those columns, please. Get the columns that are control. And so this little bit of code that's underneath that I've given you will break it out and figure out how this is working. But notice one thing here. See, it's got the equals symbol. Let me zoom in for a minute. It's got this equals control. That's the same thing we just did. Remember when we were looking how many control were in the metadata column called dex? So here's the metadata column dex. This is a different way to get it, but using the square parenthesis. We could use the $dex here as well, metadata $dex equals control. What that's going to return, this bit here, is the true falses, right? Just like we got before, and then we summed them up to find how many there were. But this thing here is the same part of it. So let's go back to RStudio and see how we're doing. I'm going to take this code and we'll explain it. We'll work it out. So, okay, so go back to RStudio, and I'll bring you with me over here. So we're let's do toy. So this is kind of a walkthrough, so we understand what's going on. I'm putting the two pound symbols or hash marks there. That just is going to make this a level 2 heading. So here we need to extract or isolate or whatever other words you want to put, or control and the treated. To compare them, essentially, right? So I'm just write this in your own words, right? So it makes sense to you. So I'm saying we need to extract or isolate or zoom in on or whatever, focus in on the control and the treated. I'll maybe put the control in quotes here or something like that, because this is the terminology that the authors used. Control, treated being the one with the dex drug in them. Okay, so first to do this, first find the column names of the control experiments. Okay, so that's what we want to do. So I'm going to paste in a new code chunk, and actually this code I gave to you, but we're going to try and explain it out. So I'm going to, when I see a lot of complicated code like this, I'm just going to say, okay, what's happening? So I'm going to comment out all these things. And actually, I'm going to comment out this one too. But this is where we're going. But first off, we're going to figure out what's going on. So that metadata equals control is the first step. We actually had that. We did that when we wanted to count that. So let's do it that way. Not meet data. I'm not that hungry yet. Metadata. And we're going to look in the dex column. We'll use the same way we did it before. So dex, and we said equals. Let's print this out to remember what it looks like. Remember, this is the column from this thing, from the metadata thing. So this is this dex column that we've just printed out here. This is this column right here. If you can see where my mouse is, this column, the second column that has control treated. And then we've printed that out, and that's what's printed here. We want to say equals, let's do control. And this returned our true falses, of course, right? So our true, false. And then we can use that as a way to look up all of that metadata file. I'm going to use that to access the rows in the metadata. Okay, so what we're going to put this in here is we're going to put this inside square brackets, meta, and then this will be the row identifier, so it's going to be true, so it's going to show me the first row. I'm going to look at all columns here to start with. So what's this going to give? So notice the comma here that I put, so it's rows and then columns in this kind of syntax here. So I'm going to have the true, so it's going to show me the first column. False, it's not going to show me the second column. True, it's going to show me the third column. It's not going to show me the fourth column. Let's try that. So what this should print out is the subset of that metadata file that is just control. Let's try it. Yay, it did it, right? So here's and I can see that printed out the first row. You see the number 1 here, so it printed out the first row, then it skipped the second row because it was false, and then it did the third, then it skipped the fourth because it was false, and it gave me the fifth, skipped the sixth, give me the seventh. So it's giving me all this. The only thing I actually want at this stage from this is the ID, this ID column, right? Here, this thing. Because this is this will be the column names in the main count data. Remember that thing that had all the actual genes and their counts? I want to use this to look up those columns, to find those columns in the count data. So what I'm going to do is I'm just going to call this control. So I'm following on. This is essentially the same as this line here. So I'll have control, and then if I access control $, now I'm going to access the first column of it, $ID. So what that'll do is it'll just print out this column. Okay, so here we go. Got the IDs. So now I can use that to look up the counts. So let me do that. Actually, it's here in this line here. So I've got, if I uncomment this, so I'm using this control ID here to, maybe we'll break this into a separate, no, we'll leave it for now. So this would be, let's add a comment for a second. Find the ID for control experiments, control samples. Then use the control ID, use ID to extract, or isolate, or print out, or do whatever. We're actually going to compute something from them in a minute. Use the ID to extract the control columns of our count data. So I'm writing these notes for myself so I understand what's going on. So this here is going to do that. I'm just going to look at the head of that now, the head of control counts, and see what that looks like. So here I've got it. So I should have 4 things. Remember, we found in the question above there were 4 control experiments, 4 treated. So I should have 4 columns here, and indeed I have 4 columns, and these should be the control identifiers. If I look at them, 508, I can remember. If I don't remember, I can look. I'm looking at 508, 512, 516, 520. Let's just make sure. Let's click on the metadata that I still have open up at the top, and I'm looking for control 508. Excuse me, 512, 516, 520. Happy days. Looks good. I could check it in code with equals and make sure, but it looks great so far. So what am I going to do with this? Well, I'm going to, how do we summarize this work, right? So these are multiple replicates of this, or they're actually biological replicates. They're different cell lines, right, in this case. So what I want to do is summarize them. How would I summarize them? Well, one easy way is just to calculate the mean across these control experiments, and then I'll go and do the same for the drug treated. I'll calculate the mean of those, and I'll compare the means. And if the means are different, then they're different, right? I can calculate the p-value for that, significantly different, these sorts of things. That's one way to compare it. So let's do that. And actually, the line of code here that I haven't run yet, this thing, I'm going to take that out and I'll do it in another code chunk perhaps. Let me turn this print off out so you can still see this code. So now I'm going to write some text now. So to summarize these experiments, these multiple control experiments. Sorry, my keyboard's at a funny angle here on my laptop where the thing is. So to summarize these multiple, and I can go back and fix my spellings in a little bit, experiments, I'll calculate a mean for each gene. A mean counts. Value for each gene. And where are the genes stored? Are they in the columns or the rows? They're in the rows, right? So I'm going to calculate a row-wise mean, a mean across all those rows. So that's what I'm going to do. Next, on the code that I give, I still have it on the pasteboard here. So it did control mean, and what it does is this control counts that we calculated above. Remember, we printed that out. That just had the 4 columns of all the count data for the 38,600 odd genes. And here I'm calculating the row sums, so it's going to sum up. It's going to sum across that row, and then it's going to divide by 4. So I think there's a question in the workbook that tells you how would you improve the code above, right? It's the 4, right? Because I'm hard coding because temptation just to be lazy here. Excuse me, and say, well, I know there's 4 control experiments, so I'm to calculate the mean, sum it up, divide by the number of them. So I hard code 4 here. What happens if I have to run more experiments, or the data gets regenerated and there's only 3 experiments, or now there's 7 control experiments? There's not enough statistical power. I need to go back and do more work here. There's another update, and then I just run my analysis script like this thing to generate my PDF report. It's going to be wrong, right? My means are going to be wrong because I've hard coded. I took this lazy choice of just putting 4 in here. So that is one of the questions in the workbook, I'm sure of, because that's a crappy way to do things. So there's a number of ways I could fix this. One would be to find out how many rows are in this thing. So let me still comment this out for a second. I want to know how I have to divide this thing. So I could just do n row of control counts. And that would tell me how many row, sorry, I need to do n call, not n row. pardon. This would tell me how many columns am I summing across, and this will tell me 4. So I could put that in here, right? This call. This would be a better thing to put here. So I'm dividing it by that. And this will be much more robust. I could, of course, if I looked at the documentation for row sums, I would maybe find out, well, let's pull it up to make sure we understand what this is doing. So I'm going to push F1, function F1 here. It's this first one. Then after it says from row and column sums and means. Oh, I can actually just calculate the mean here. There's a function called row means. So I could just use that as well. That would be even easier, right? So I could do that. So let me just replace this by row means. And I don't even need to do that division here. It does it all for me. So I'm going to do that and have a look at the answers to see an answer. And I've got it. Okay, happy days. So now what I've done is I've gone and I've extracted just the mean columns from the count data, and I've calculated their mean. Okay, that's one way to summarize them. Of course, it's going to be maybe not the best way, but we'll work with it for now. Okay, what do I want to compare that value to? Well, I want to compare it to the means for those samples where we had the drug, right, and see if there's a difference. So I'm going to have to do the exact same thing for the treated. Okay. And I think in the workbook it asks you to please try and come up with this code yourself to try and do this. I'm going to do this too. Treated. So here, let's code it up again, and maybe we'll. And I think if you like that tidyverse approach, we could do it that way. There's always multiple ways to do things. We would use the filter, dplyr function to do that too. You could certainly do that. I'm using the BSR approach because it will work everywhere. It's the basis of how all these things work anyway. But it's a little bit more complicated. But if you understand this, it's kind of easier to understand a lot of other approaches to do it. I'm kind of doing it the hardcore way here. Let's keep with this way and be consistent. So what I'm going to do now, I'm going to have to look in the metadata again. And it's tempting to, and probably the right thing to do in this case is just go up and copy and paste and change the word control for treated, right, in this case, but I'm just going to do it here. This is the metadata dex. I'm going to say equals treated. I'm going to make sure this looks right. So this should be false true in this case. I'm going to use that to find all of metadata. Okay, so this will print out step by step. I'm checking my work as I go. So that's printing out just the treated samples from that metadata thing. And I just want the ID column. So I'm just going to do ID here. And I'll print out the IDs. So let me put that here. I could do treated ID here. I would store that. And then I could go and do count data. Counts. And then I could use that as the treated.id. Let's have a look. So I'm going to do head, so I'll just print out the first 6 rows of this thing. And here because I didn't spell it correctly, it's giving me an error message. So here's 6 rows, and I'm still going to, even though I know I'm pretty much on the right track, I'm just going to make sure. See this 509, 513, 517, 521. I'm just going to go and make sure. So I'm looking for 509, 509, 513, 517, 521. Perfect. Got it. Right. I'm checking my understanding as I go. So instead of doing the head of that, I'm just going to do row means. And then I'm going to store that as, what did I call this one? I call this one control.mean, so I'll call this one treated.mean. And I'll print out the head of it. Just the first head will by default give you the first 6 values rather than printing the whole 38,000 odd things, right? You'll just go on and on, make page and page and page and page in your PDF documents. You don't want to do that. You just want to have a little peek at it. Treated.mean. I'm going to run this chunk again. I should see these values. So I notice here some things, right? So the first gene has a big value, then the next gene has 0, right? No counts, no data. Right? We actually don't know anything about this gene. And there's another one, the last one here, the 6th one, also has 0. Anyway, but it is what it is, but we'll come back to that. Also, the second gene up in the control has no data, so there's definitely nothing we can say about that gene, right? It's got no expression when you don't have the drug, and it's got no expression when you do have the drug, or no detectable reads mapped to it, at least. Okay, so we're definitely not going to be learning anything about that gene in this experiment, so we'll probably need to filter those out, right? Let's compare them. One way we could compare them is just plot. And it's always good to explore our data and plot. So let's make a quick plot. Data a little more. So I'm going to plot here. I'm going to use base R functions again to plot rather than ggplot in this case, but I'm just going to do plot. And what I want to do is I'm going to plot the control mean versus the treated mean, right? Let's see if I'll make sure I did it. Yeah, okay. Let's do treated mean, and then I'm going to do control mean. I'm just going to plot x versus y. So x is the, why can I not spell when I don't look at it here? I should use the tab key to help me here. I'm typing in these funny angles, so treated and control. So this will produce a plot eventually of these 38,000. So there's 38,000 points in here, believe it or not, of the treated along the base and the control. And let's think about it for a minute. What would this be? Well, if there was no change, right? If the expression level whenever you didn't have the drug there, and expression level when you did have the drug there would be the same value. So they would lie along the diagonal. Right? They would follow, there would just be a straight line going from 0 all the way to whatever the max. And you can see that what was common with this, there's some genes, a few genes that have tons and tons of expression, really high counts, and lots of genes that don't have very many counts, right? So this is data that is highly skewed, right? This calls out for a transformation, like a log transformation, as we'll see. But also, hang on a minute, like wasn't there 38, remember when we did how many genes were in this? This 38,000 genes, this 38,000 points. This looks like, what, a couple of hundred, if that, right, points that I can see. It turns out, that all the genes, or the vast majority of them, are actually all on top of each other. They're all down in this region here. This is where a lot of the action is, with low counts in both control and treat. Now, the nice thing that I do see is that there is some spread, right? There's some genes that are up above the diagonal. That means there's more of them in whatever I plotted on that axis, control. So they're going to be have more expression in the control if they're up above the diagonal, and then if they're down below the diagonal, if they're down here, they're going to have more expression in the treated, for example. All right, so there's some difference here. That's good to see. If this was just a straight line, I'd be like, okay, well, next lab session, please. Right, next thing. There's nothing to see here. Move along. So there is something worth exploring, but I can't see all the data because it's heavily skewed. So let's do a log plot. Let's change this code to do a log transformation, which is one of the most kind of important transformations we can learn when we're analyzing data. It's so useful to do this when we've got heavily skewed data, we've got lots of signal down here in these small values that we want to draw out spread over such a large range. So a common transformation is the log transformation. Let's make a better plot by changing to log plot. This will show us more of the data, log versus log. So how do we do that? Well, I can actually do it with the plot function itself. So I can copy this code from above here. I'm just going to copy this, and I'm going to look at the help page for plot and wherever this is actually plot.default that goes off, and see if I can change this to a log plot. I'm going to pull it up. You could type in the console $plot. I'm going to just hover my mouse over it and push F1 key on my keyboard. That'll bring up the help page. So here, behind my head, it's a bit squished in, so let me draw it out in a bigger window. I'll pull it up here so we don't squish it in too much. Generic plotting function, there's these arguments. Go see plot.default, please, it says. So I'm going to do that. And I'm going to look at these arguments. There's one called log. Right? There's X and Y type. Remember we saw that before. If you want a line plot or these sorts of things, you can have the limits of the axis, and there's lots of other ways to argue it. But there's one called log. That sounds like it has something to do with log, right? It is a character string which contains X if the X-axis is to be logarithmic, and Y if the Y-axis is to be logarithmic, and XY or YX if both are to be logarithmic. Okay, so basically this is telling me write the word log as an argument equals whatever axis you want to be log transformed, and I want both, so I'm going to do this XY. Let's try that. I'm going to close this. I'm done with it. I'm going to go back here and argue with it and say log equals x, y. I'm going to plot it, and now I'll have and I can add labels and say x lab and y lab. Oh, yay, I've got more points, right? That looks like a lot more points. It's definitely made my data much more visible, right? It's spread it out. I can see that there's tons and tons of points here. There's still a lot of overplotting. I could do transparency, I could density shaded, all these things, but it's much more reassuring to see my data, right? And I can see that there's some spread above that line, above and below, that we're going to dig into. Before we go and delve into this, let's save our work thus far in one object. So I'm going to take that control.mean and treated.mean and save it into one object that we can work with and keep building on. So I'm going to make sure I'm not straying too far from the script or that we've missed some questions by going back to our web instructions, our web walkthrough here. So we've kind of done this. We've question 3: how would we make the above code more robust? That is, not hard codes the number to divide by, use row means, or determine the number of things to divide by from the data itself here. And then we've done that, follow the same procedure for treated. We did that together. And here it says, please combine, for bookkeeping purposes, into a new thing called mean counts. I'm going to make a data frame that stores those two things that we've just calculated, the means for the control samples and the mean for the treated. So I'm going to follow my instructions and do that. Let's see what else where else we're going here. We could use call sums to find out how many there are for those. That's fine. Here's the plot that we made. Here's the logarithmic plot that we made. You can see here that I'm adding the X lab and Y lab to actually label the axis a little better. It says, so log transform, there's more in the hints if you talk to them about why log is a good transformation to do. We often use log2 rather than natural log, which is what's being used there for that plot, or log10 that you're probably most familiar with. Log2 is is going to be is really useful and easier to think about for simple-minded folks like me. Right? That if we have a doubling, well, let's check. We'll use this log 2 of the mean counts and store that. So, and then we'll come back and talk about the good properties of log 2 here that make it a bit easier for me to deal with. So let's go back to RStudio here and do what we said, which, where am I? Okay, so store results to date in one data frame. This is just copying and pasting the code from called mean counts. Okay, so what we're going to add onto this mean counts is the log2 fold change, right? That's what we're after. So this, let's have a little play here with log2 just to make sure we're, so if we had, like the way we plotted one versus the other, if we just put one value and divided it by the other value, if they were both the same, it's going to be 0, right? If we had 20 counts in one sample and 20 counts in the other, right? That's just a fraction. That's going to be 1. Right? 20 over 20 is the same as 2 over 2, which is 1, right? So that's going to be a value of 1 if you do this. Here it's going to be. So if we did the log 2 of this, the log of 1, the log 2 of 1 is going to be 0. Right? So log 2 of 1 is 0. So if we have a log 2 fold change, right, one over the other, control over treated or treated over control, depending upon what way you want to do it, it's going to be up or down in respect to the way you do your fraction or the way you do your division. If it's the same in both experiments, right, if it lies along that diagonal in the plots that we just did, you're going to have a log 2 fold change of 0. Now, if you had more in the first experiment, say we had 40 here, so double what we have in the second experiment, that's going to be, obviously, if you did it without the log 2 here, that's, we all know what this is, right? This is going to be 2, so it's double. Okay. Now, if you do the log 2 of that, that's going to be 1. So a doubling in this log 2 units is going to be plus 1. If it was the other way around, so 20 and we had 40 on the bottom, this is going to be minus 1. Okay. So in these log 2 units, if you have a log 2 of 2, right? So if this was 80 here, it's going to be minus 2. Okay, so we're going to use this. If we see a minus value, we're down that many folds, right? Or if we see a plus value, we're upregulated. So we're going to do a log 2 transformation here because it's simpler to think about than natural log this thing, because that units are going to make it much easier to see. If we have a log 2 fold change, we've quadrupled. Right? We've got that many times. A log 2 fold change of 1, we've doubled, either up or down. If it's a minus sign in front of it. So let's do that. Oh, there's one thing before we do it. Let's think about, well, what would happen if, remember we had, when we looked at the head of the mean counts and the head of the control counts, we actually had some 0 values. So there was actually no data for one gene. So that would be like we had 0 here. What's this going to be? Well, 20 over 0 is going to be 0. And what's the log of 0? Well, it's not good, right? It's actually infinity, right? To infinity and beyond. So it means we can't say anything about this kind of data, right? If we have no data for it, we're going to have a log of infinity here. Right? So that's what. Let's keep that in mind, because we actually can't say anything about those genes if we do it this way. So let's do it. Actually, I give the code back on the web page. I'm just going to go there to make sure I do it the right way around. I put the right treated or control on the top of the fraction or the bottom of the fraction. So I'm taking the log 2 of the treated over the mean, and I'm storing it as this thing called mean counts. let's make sure you can read it. This, I'm just storing it as this new column called log2 full change. Okay, so let's take this and go back to RStudio and put it in, and we'll see that we're nearly finished this hard section. So let's take the log2 full change. Okay, so this is wrapped here because of my windows. It's small. So now we're going to have, oh yeah, I haven't run the mean counts yet. There's mean counts. But I do that. So now I have my one single data frame that has the values that I calculated, the row means of the control samples, the row means of the treated samples, and now it's got my log2 full change. And here it's telling me that it's down in this one, and that it's up very, very, very slightly in this one. Okay. And I've got these infinities in here because I have a 0. And of course 0 over 0 is not available. This means not a number in this case. So I have these weird answers in this log2 full change. Most are looking good, but anytime I have a 0 count here, I'm going to have trouble. I'm going to have these values. And actually, things like this second gene here, we're not going to do anything with it anyway because there's no count data available for it. So we're going to have to filter these out. There's no point doing a comparison or a hypothesis test or something and reducing our statistical power here by comparing something that we don't know anything about. Right? So we're going to filter this out. So to do that, we're going to need to find any genes that have 0 counts in them. That's this step. We're going to have to go and figure out these and just remove them from our data set. So how do we do that? We're going to use that same, let me write some code. So there are some 0 counts, genes, in our experiments, our samples, leading to full chain values. We need to remove these. Before going further to test the significance of the differences in these means, for example, with statistical tests. I'm going to try and fix my spellings. Okay, so how are we going to do it? Well, I've got these 2 columns, or we've got these 2 columns, control.mean and treated.mean. So I could access these. Let's just work with these little first 6 columns first 6 rows rather than the whole 38,000 odd, because that would be a little confusing. So let's try, so this is our mean counts data. Let's take the head of it I've already printed up above. I'm just going to print the first 2 columns. So here it taxes first 2 columns, just go 1:2. So what this is going to print out will just look the same as above, but it's not going to print the log2 full change. It's going to print this column and this column, the first 2 columns, and the 6 rows. So that's that little subset. And what I want to do is find any gene where there's a 0 in it. So if I did equals 0, and let's just do take the mean of this out here for a second. And we'll do here, just to work on the first 6 rows, and we'll do 1 to 6, just to keep it a little bit more simple for us to see what's going on. And I'll let me do let's just do what I did before, the head of counts 1 to 2. Okay, bear with me for a second. So this chunk here was just what we had before, so I've got that showing. And here what I'm asking is, now tell me where the zeros are. So hopefully we can see, oh, can I fit this all in? Kind of not. Okay. So it's going through and I'm using this equals equals, which again, we used before there, and it tells me that that true is where the equals equals comes out as the thing on the left being equal to the thing on the right, so there's going to be 0. So this tells me true, and if I go back up, it tells me yes, that is a 0. Yes, this is a 0. So I'm seeing the same kind of structure. See the true, true, and then down here there's a true as well. Let's have a look. Yep, that looks good. Okay, so I found the 0 entries, but I need to know where they are. Specifically, I need to know the rows they're in. There is a function that we did use it previously, but it might be back in the mists of time, called which. Let me just show you what which does before I type in this thing out. So let me, we'll leave this thing here. Let's leave this alone for a minute. Undo. So if we had a vector, true, false, I'm just using the capital T, F, T here. So this little vector that I print out will be true, false, true. If I wanted to know where the true values are in this vector, I could use which. And that will tell me the positions or the indices. It's the kind of terminology that the early folks use for the positions in it, like the first and the third are true. So it's giving me this little vector saying, yeah, first, third. Right? So if you added some more F's in here just to show us, it's going to tell me that the first and the fifth now are true, right? So that's might be useful. Let's see how this would work. Okay. So if I remember, I have this table here that has the trues in it. If I do true in it, what I really want to know is what is the second row that has a true in it and the sixth row that has a true in it. Okay, let's see what the which gives us. So I'm going to wrap this in this which call. It doesn't really give us what we want. What it actually tells us is the second, the 8th, and the 12th. Where the hell does this come from? Second, 8th, and 12th. Well, if we count down, it would be 1, 2, yes, that is 0, 3, no, 4, no, 5, no, 6, no, 7, no, 8, yes, 8, so 8 should be in it, as long as so 2 and 8 should be in it, 9, no, 10, no, 11, no, 12. So it's actually given me the positions in the vector. As if it took this little table and flattened it into a vector where it goes down the column, and then once it's finished that column, it goes up to the next column and adds it on and keeps going like that. Now there is an argument to which will be helpful to us. There's a which argument to which will be helpful. Okay, I'm going to stop. On this argument, we could look at the help page. My window's a little squished in here right behind my head, but I'm going to pull it up here. I'm going to push F1 to look at the help, and I want it to argue with me. So this one, and let me pull this up. So here, the second argument of this document is array.end or rrr.end. Logical. Should array indices be returned when X is an array? This terminology might be a little full-on nerd-tastic, right? But this is essentially what we want to do. Let's play with it and see if we can figure out what it's doing. So I'm going to do here this argument, array.end equals true. Let's see what this returns. It actually returns row 2, column 1, row 2, column 2, row 6, column 2. Let's have a look and see if that's the case. So row 2, that would be this row, row 2, column 1, yep, that's the first column. Row 2, column 2, yep, that's this one. Then row 6, column 2, so this is row 6. This is row 6, and this is column 2. Boom. got it, right? So what we want from this is just the rows, actually. We don't care whether it occurs in the first column or the second column here. If it's a 0 in it, we're going to have these funny effects from the log values, and we're not going to be able to say anything about the gene anyway. So we're just going to look at the genes, the rows, for which there are zeros. So in this case, I just care row 2 and row 6 have this problem. So I want to know the rows, so I could actually, here I can do row like this, and that will tell me just the rows. And there's multiple ones in here, see, multiple 2, so I want to make that unique. So I'm going to go and do unique like this. And this will tell me 2 and 6. And I'm going to do it for everything. I don't want just the first 6 rows anymore. I want to do it for the entire data set. This is going to print it all out, so I probably shouldn't. Let me save it as 2.rm, little vector called 2.rm. This will be the rows that we actually want to remove, and I'll do a head of 2.rm. Now if we did it, so this is going to be all the rows 2, 65, 60, 73, 121, because it goes down the columns first. If we went further on, you would see there's a 6 in there as well. So let's have a little look at this. So here I'm going to do mean counts, and I could use this to access the rows, 2.rm of the rows. So this will print out. Maybe don't want to print it all. I'll just do head again because there's going to be a lot. So in here, when I print this out, this should be genes for which there are 0 values. Yep. You can see all these have 0 values and funny log2 full change answers. So I've got it. Right? I've accessed the ones that are to be removed. Now, how would I print those that I actually want to keep? Well, there's a couple of ways I do it. I could have a vector called 2.keep where I say not equals here. That's one way to do it. But I can also just flip this thing by saying minus in front of it. So the minus here, what that's going to do is give me everything but these positions. So when I print this, I'm going to go back. I should have my first gene. And remember back up here, I shouldn't have this second gene, the 005, but I should have the third one, 419. I also shouldn't have the sixth gene, the 938. So I should be missing the 05 and the 938. Let's have a look. So there's the first gene, and then it goes to 419. Let's have a look. Yep, that's looking good. And then I should have 460, and that should not be 938. There's 460, and then it goes on to the next one. Happy days. I've got it. I've filtered out the problem values. And I've got my fold changes and my treated and my log2 values. So I'm going to pause here for a second. I'm going to save this results to date. So let's save our work. Save our work to date. This will be data frame, new data frame. All the 0 count genes. Let's see what I call this in the workbook. I can't remember. So if we go back to our website for a second. Here, we're going to call it my counts. Okay. Then we ask how many genes are up and how many genes are down at this common log2 threshold of 2. So how many genes have 4 times as much expression in one condition as the other in this case. So we're going to save this as my counts. Go back over here. and save this as my counts, and then I answer those questions. So what we did, the last little steps, is a little bit of a step up. It's a little bit more confusing than some of the code we've been working on. The dplyr way is actually probably easier in this respect for the filtering, but I'm emphasizing this base R way to do things because it's the foundation of all these approaches. So let me run this. So we have our my counts. I can look at it. There's how many genes left that we haven't filtered. I could look up here, 21,817 rows in this. So what's the size of the human genome? 20,500 something, right? So it's 21,000 odd genes, nearly 22,000 genes. That's what we're looking at here. There's a lot of pseudogenes in that full data set and other things, and splice variants and whatnot. So we're down here to 21,000 and 22,000 genes. And we want to ask, how many, so a common threshold to begin with. Threshold is log to full change of 2 or more. So if it's less than -2, it's going to be reduced. If it's above +2, it's going to be up. Right. So let's look at this common threshold. Let's look at this common threshold of log 2 threshold cutoff. +2 for upregulated, and -2 for down. How many genes are up or down? And you can put these in this up and down in quotes. Obviously, we're missing a step where my inner statistician is screaming at me, right? Because we've got a difference in the means, doesn't mean that these are really, it could be changing a huge amount. Whenever you add the drug, the expression could be all over the map. It could be all over the place, and then when you have to treat it, it could be just down here, right? It doesn't mean it's a significant difference, right? But let's go with it anyway for now. Consider just the log 2 fold change. So I'm going to try to figure this out. So this is my counts, $log 2 fold change. And we want to know how many are up. So log 2 greater than 2. This is going to give me this huge vector, true, false, false. I don't want to print it all out right here. How would I find the number? You guessed it, sum, right? Because these are ones and zeros, the trues and falses. Sorry. So this will be 250 genes are upregulated at this threshold. How many are down? I could do some of my counts. $log 2 full change less than -2. And it tells me 367. So slightly more downregulated than upregulated at this threshold. You can saw that in the plots too, actually, to an extent. So let me pause here and ask you, great, do you trust these answers? So I want you to think about it. Right? We've done quite a lot of work. What have we done? Well, maybe we'll have a look and check how things are going. Maybe pop back here as well. So I'm going to there's a site. So did I not? I didn't post the announcement today. Oops. Okay, let's run with it anyway. There's some good questions, answers here. I haven't seen some of these. Thank you very much, folks, for posting the questions for our industry speakers. We'll get to those and we'll talk to them in a separate video a little bit later. I'm going to go back here and go to class 9. This will hopefully respond or feedback on folks who posted the question. Okay. So that's a funny pose. Let's stop there. Things seem to be working. What I'm going to do then is just plow on in here. Apologize for not starting the Piazza forum discussion on this. We're going to actually do this the right way. We'll finish off here with a DSeq analysis of this. We'll see that it's much easier. What it does under the hood is calculates this log2 fold change, but it also does the statistics. That's the thing we're missing. Right? We haven't actually said, is this difference in the means that we're calculating significant? So DSeq2 is going to do this. It uses negative binomial kind of statistics that these things, the same as we would have the binomial where we can flip a coin and see these kind of statistics. That's the kind of distribution that we'll use, and we're going to do this DSeq2 analysis. We're going to call up our DSeq2. We're going to get this table of results where we have our log2 fold change, as well as the missing p-value that we're going to do. So let's go and do that. We're going to take this little chunk of code here and set up our DSeq2 analysis set. So this is in section 4. I'm going to go back to our studio here and say, well, leave you to answer the remaining questions there. So I'm going to do section 4. DSeq2. So let's do this the right way with statistics. And we're going to load up the DSeq2 package by the DSeq2. And when we load this up, it's going to give me all that text in my document. I'm going to then use, and this is coming from we're going to take the recommendations of the DSeq2 documentation, the vignette. So it needs this, it's calling this long-winded function name, a DSeq data set from matrix. That's going to take our count data, our call data, and then this third one I'll explain in a minute, the design of our experiment. But this is very common in bioconductor functions and packages and uses. This is it wants these special object types to store the input and also store the results. You'll see that we will actually write our results back to the same object in a minute. So we're going to run this. What we're passing here is counts. Remember, counts was that thing that we read in was read CSV, our count data, and then our call data, that's the metadata. And then we're going to tell DSeq2 right at the start here, what is the setup of experiment? What do you compare, what's the design of your experiment? And we're just going to look and say, we always start with this little tilde. It's going to say, go look in the metadata at the dex column. Remember the dex column, when we looked at it, that's just the telling us which is control and treated. Remember we did this manually and pulled out which were control and which one are treated. You can also use cell type and other more complicated setups. We're just going to keep it simple initially and just say, please compare control and treated. we're going to set that up. dseek2 will go and do that. We can have a look at this object. Again here, it tells us that it's a data set. It's got these 8 things. It's got the columns. There are 8 of them, etc. Okay, and the metadata is the ID, dex, cell type, geo. So now I'm going to go and run. I'm going to store this as a thing. Oops. Seek DDS. So I'm going to do that, and then I'll put that DDS here. This goes and runs the DSeek analysis pipeline. It does everything we did and quite a bit more on the statistics side, as we talked about in our videos. It does the multiple testing, correction for the lots of hypothesis tests that we're doing. So we've done it. It's done all these different steps that we talked about, and it's going to give us our results back inside this main DDS object. To get the actual results, so to get our results, we're going to so we've got, I'm going to use, call it res, just short for results, give myself some room here. And we use the DSeek function results, DDS, so get me my results please from this object. Let's just print it out. And it'll be quite big when we print it out. Probably don't want to print the whole thing, just do the head of it again. If the code's running, I'll leave it going here for a second. So it's got the base mean. So this is the mean across all our columns, all the samples in this case. Then it's got our log2 fold change. Remember that second gene that had 0 counts? Look, see again, it's got 0 here. This is, and there's other columns that's used for calculating the p-value, right? So if there's the p-value again, that second gene, so it's wrapped around at 0.005. It's the same here. DSeek doesn't do anything with it. Doesn't have 0 counts. Doesn't have the log2 fold change. Doesn't have the p-value or this Benjamin Hushberg adjusted or corrected p-value that will be larger than these conventional p-values. This is for our multiple testing issues that we discussed in our videos. And you'll see that the 6th one doesn't have data 2 if we got it. So it looks like we've got everything we need here. Got our log2 fold change. I'm going to save these results to a file. So let's save to file. So we can just read them in next time if we want to do anything with them. So I'm going to do write.csv. We could order them by the p-value or the combination of the fold change and the p-value. We could add extra annotation like the gene symbols and things like that. I'm just going to do file equals DSeek results.csv. I'm going to write that out. I might need to change it to a data frame first. No, got it. So here's the object written out. I can see it right behind my head here. Over here, this thing's written out a CSV file. Here with my results, I could just read that in next time if I wanted to do something for it. But what we've got is our results. We can do a summary of that res object, and it'll give us some details about it. So it's telling me at the log2 fold change of 0, which of course we don't want that. We want higher values, and the adjusted p-value of 0.01. We have this many up and this many down. You can argue with this summary. You can set an alpha value, for example, or a log2 fold change. Alpha would be 0.05, for example. Then it would print print a summary of what's going on, how many genes are up or down, and you can do it with the log2 fold change as well, and find out what proportion are up and down and the like. But what we've got is a big damn table of results. Right? If we click on this results thing, it actually doesn't show us anything about it because it's this dseq results class object. So I'm going to change it to a data frame here, so we can view it. So we're going to do as.dataframe inside a code chunk. And I'll do my results here. So let's just call this res.df. And then we could click on this or do the head of it or view it. So do head res.df. It'll print out just the start of it here. I'm going to, if I could see this, let's put this onto grid view. I can actually read these names. Doesn't have, because I'm too squished in. See, so there's our res.df. So we've got our big table right here. We could have typed just view res.df. So we've got our p-adjust, our p-value, our log2 fold change, and it goes on for all these, all the data that we've got. Okay, great. So in isolation, this is not that helpful to me because I have no idea. Well, first off, I would find the genes that are up and down here, but this annotation here is not really that helpful to me. I want to annotate it by actually giving it its gene name. So we're going to do that. Let's add annotation to our results. And this is the next section of our lab sheets, to add annotation to these things. So I'm going to go over to that section here. If I can get my mouse to follow me to where I want to go. So here's the web page. And we're going to do section 5, adding annotation. So here, there's a few different ways we could add the gene annotation back on. If you had a file from UCSC or Ensemble that had the gene names and whatever other identifiers mapped, you could merge them. The merge function would do this for you, or the left join function from the tidyverse would do the same sort of things. I'm going to use the bioconductor approach, which uses two packages, annotation DBI and which will allow you to add annotation to these data sets. And then a data set for humans, see the org HS. HS means the human here. So I'm going to load these up and add this annotation on with the map IDs function. So it's going to take that human database big table, and that's going to say we're going to take the row names of our results. This is our results from dseq. Remember, the row names are those ensemble genes. And then I'm going to just add on, please, ensemble, tell them that they're ensemble form, and add on the gene symbol in this case. Okay, let's do this together here. I'll take this text and bring it back over to our studio. Here. And paste this in, banana annotation. If you don't have these packages, so the first time we do this, you're going to have to install these packages in your console the same way we did. Remember the BIOC manager function? I'm just going to load them up here because I have them installed already. And now I'm going to look at the columns of the org. see what we've got. So the columns will tell us this is all the different identifiers that we can map to or between. So we can map from ensemble IDs like we have, right? There's the ensemble identifiers. That's what's in to anything else like Omen or Go or RefSeq. These are all Uniprot. Some of these should look, most of these should look pretty familiar to you. And we're going to map to gene symbol over here. We'll also map to entrees IDs. Remember, entrees was that NCBI thing for looking across all of NCBI data sets. So we're going to map to gene symbols, entrees ID here. So we're going to use the map IDs function. We'll use the main map IDs function to add different identifiers to our dataset. And we need to do this because we need to understand we need to know which genes are in our dataset, but we also want to be able to use pathway analysis methods and gene set enrichment methods. And they all use different database under the hood, different identifiers. So we need to kind of translate between these languages, between these different database identifiers. So we'll go and do that. And I'll pop back over for a second and get the starting code from here. So we're going to add, in this case, I went back to the web page, and so we're going to take the org HS. So this is this data set for humans, and we've got our results are in this thing called res, right? That's from our DSeq results. The actual gene names in that are the row names there, if we remember. Up here, if I scroll back, these are the ensemble identifiers. Those are the row names here. Okay. So I'm going to then tell map IDs that these are ensemble. This is the key that I want to use to go and look up this thing, and they're an ensemble from it. And I want to add gene symbol, please. And then this last argument, multivals, this says if there are multiple matches within these different databases, different splice frames, different identifiers, you can often map to multiple other identifiers in other databases because they're all set up slightly differently, different forms. We're just going to take the first one. So this is going to add a new column called symbol to our data set. We can do that. And then we have a look at the head of the res here. We should have a new column in it called symbol. So at the end, symbol that has our gene symbol. So this first gene is TSPAN6, TMND, etc., etc. Okay, FGR. These are a lot more useful in those ensemble IDs. We could also add other things. So let's go and take this and add some different types here. So more mutation IDs. So let's add, instead of symbol here, so we'll change this symbol to the different one. If we remember that when we printed out the columns up here, we can map to any of these. So I'm going to take, let's take, okay, well, yeah, we need entrees ID, need this one, take this one. There's also gene name. Let's take gene name because that would be actually something we can read. Right, so I'm going to add gene name here as opposed to the one before, and I'm not going to map it to the symbol, but I'll just call it let's call it gene name. That, and then I'll have do the head of this data set again, just to check that it's been added. So when I do this, it'll add those, and then I can go and look at the last column, which in this case has the names of the gene: tetras, baron 6, tentomodulin, right? Chromosome 1, open reading frame 112. So not a FGR, proto-oncogene, Circ family, tyrosine kinase. So a lot more useful. I'm beginning to learn something about these genes, for example. So I'm going to do one last one, the entrez ID. So let's take this. We'll take the whole thing actually. We'll go add it here. We need entrez ID for the tool that we're going to use next because it speaks entrez language. Actually, paste that in and we go here ENTRC. And here we want, instead of gene name, we're going to map it to something else. Entrez ID. And then if we did our head of this, we should have a column after with these entrez IDs. So these are these numbers, right? Not very easy to read, but they mean something to the entrez database and any tool that speaks entrez, right? That's why we're adding these things. So the next tool that we're going to use to add a little bit more annotation to it is this pathway analysis approach. And we talked about this in our videos, recorded videos for this course section. But the idea here is, so I'm going to go back to our web page and briefly scroll down. So there's the annotation. We're going to add, the volcano plot. Yeah, let's do that first because that'll focus in on. So what I was mentioning is we were going to go and take our subset of genes and map them to pathways here. Okay, so the next step would be to do this pathway analysis and find out which genes are regulated, and we'll see results like this. So here's the KEGG pathways. KEGG is a database of pathways, and we can see here that these genes and these pathways that are lit up, these colored genes, like here, they affect airway inflammation, as you can see right above the head if we zoomed in on this thing. So this MBP is down-regulated, so it's down-regulated, which of course is responsible for promoting airway inflammation. All these other genes, interleukin-4, IL-4, all these other ones that we can see involved in these kind of processes. So we could start to get a much deeper level of initial insight into what might be going on here by doing this pathway analysis. But before we do that, we still want to, we've got this big damn data set, right, of genes, and we've looked at fold change, so we've made it smaller based on those that change a lot. And we've also got the statistical significance, the p-value of that difference between samples. But it's still a big data set. So let's see how big it is and try and do a summary figure if we did it before we go to this pathway analysis. And we'll just finish off briefly with this. So we're going to go and make a volcano plot first. So these plots, or volcano plots, they're kind of, they don't really look like volcanoes first off, but they're a common summary figure, and there's a number that we can try and make here. But we're going to do, what we're plotting here is the fold change, the log2 fold change, as you can see here. As the x, so that's going to be along the bottom, so that's the fold change here. So 0 means no difference, it's exactly the same value. And of course, if we go this way, if we go into the minus of the fold change, that means it's down-regulated. If we go in the plus way, it's up-regulated. So the further away from the middle, the more the change, right? And then we could just plot it. Let's actually plot it against p-value and see what that looks like, and that will motivate us why we take this funny unit here. So I'm going to go and do that. I'm going to go back to our studio and make one of these volcano plots. Take this text just to add some context to my R Markdown document. I'm going to go here to our studio. And before we do our pathway analysis, we'll go and do this volcano plot. This is a kind of summary. Oh, excuse me. Summary figure of our work, of our main results. So we could try and plot, so the two main columns in that data that we got were the log2 full change and this adjusted p-value. So if we just did a plot of so our results, and then we had this log2 full change, and then we did our results, and we did this p-adjusted value. So we did this kind of plot, right? That's just plotting those two columns. We get this strange looking figure, right? So each dot is a gene again, each one is a row in this thing, and the further away, the bottom axis is the further away we are from being the same, being 0 here, the more up or down regulated they are, respectively. And then all these values that are peaking up here, they have really high p-values, right? What do we know about p-values? It's the small ones that we actually care about, right? The traditional 0.05, that's all way down here hidden at the bottom, right? So we're going to use log to draw this out again, right? Because we're focusing in down here on these small values. We'll do that. So let's just take log of the p-values rather than this one, because we don't actually care about those high values. We really want the small values. So I do log of this. I get this kind of upside down volcano here. So these are all, this would be the small values here, right? The small p-values. This would be those that are closer to 0 would be down here. This would be all these points here. This is kind of difficult to look at, and it's kind of upside down from the way the world thinks about it. So I'm just going to put, and these are minus values. See, they're 0, minus 50, minus 1, because they're all 0 point whatnot, right? Less than 1, remember the way the log works. So I'm just going to put a minus here. And I'm going to put this, and we get our, albeit rather dull, all colored in the same black color, volcano plot. So we've got our log2 full change plotted on the bottom axis here, and on the side axis we've now got this minus log. And we're doing these shenanigans, taking log of it, because we really want to focus in on those small values. And then we're doing the minus because we want those to be higher up on the plot. So it's just for our visualization benefit that we're making this transformation on the p-value. But what it means is we're really focusing in on things that are higher up, the points that are at the top here of this figure, and also those that are further away from 0. So in the workbook, we go through and we can add color for to highlight the subset of points, for example, that are more than 2 log full change of 2 or more, for example, right? We could do that simply. Let's do it to start with. So if we have a color vector, let's do my calls. Let's make it a little color vector, and we say let's do its rep. We'll do gray. We'll make everything gray initially. We'll do row of gray. So this is going to be, what this is just going to be, if we just print out my calls, this is going to be gray, 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 gray. Right, so it's gray, gray, gray for as many things as we have, more than 60 rows of gray here. Okay, don't go there. This is gray. So if we just add color equals my calls, every point in our plot is going to be gray. So we'll have an even worse, even more dull plot than before. So you see everything's gray. I want to just say my calls, let's say my calls, well, what we want to do here is we want to change the color of some points to be, let's make them blue, for example, if the log2 full changes. So res log full change is more than 2, let's do that. So I'm going to say res log full change is more than 2. Let's make these points blue. This is going to color the top half blue. So these are the points that are, or the genes in this case, that have that are upregulated at this level. Okay, if I wanted to do the downregulated ones, I could do the absolute value, ABS, anti-lock brakes, or absolute. So this would be the -2, +2. This would cover both sides. I could do it separately, another line. Here these are those that meet that criteria of the log 2 fold change. You could add some lines in it. I could also draw a line across for the p-value, those that meet our whatever your criteria for your p-value is, 0.05 or whatever, and color them up, and then we could start to look at what these genes are, because this is the subset of genes that we want to focus in on. These are the ones that are both change a lot and also their change is significant. Okay, so we could do that. There's also in the workbook I show a ggplot based package that will make these plots quite conveniently for you. Let's have a look back at the workbook. It's called Enhanced Volcano. Is the name of the package, and it makes reasonably nice volcano plots. So here you go through the code to make these plots, add the lines and change the x-axis and the y-axis labels, things like this enhanced volcano plot. It's from Bioconductor. You need to install it. I'm going to, just like all ggplot, it wants it to be a data frame. As we've said, we can do this. Let's see what it looks like. If we go back to R Studio, so we'll try our ggplot version. Paste this in. So it's making a data frame to work with, because ggplot works with a data frame. It's telling it that we're going to add some x and y labels here, and then point labels, this lab for the labels. If we run this, it's going to actually load up our labels as well. So we see that here we have the subset. We have those that don't meet our criteria for. It's a bit squished in here, but you can see that those that don't. Let's run it in now. Console, so we can open it up over here. Okay. So here's my plot there, because I'm messing around with all the figure margins here. Let's see. Okay, let's not worry about it for now. It's appearing eventually. But this plot, let's make this small again, is telling us that excuse me, that it's gone back to the funny zoom, but it would label up those points. Change my zoom some. I'll close this and make it. Hide that, of course. Well, if we get this plot working in a second, let's try. See what I've broken here. Okay, so I'm going to demonstrate something that would be useful, as I've kind of messed around with it's doing lots of things here and eventually ticking on, and because I click so many times and then it's running it multiple times, so you can see here that it's off doing something. I am getting a bit tired of it working like this underneath the surface, so I'm going to go and quit my R session and restart it. You see this session up here in this window up here. So I'm going to do session terminate R. So this one here. So this, you'll see my R brain, and it says terminate R will cause your R session to immediately abort. Active computations will be interrupted once the source changes. So I'm going to, before I do that, save my document. So I'm going to do session terminate R. And actually, I'm going to do session restart R. So you can see it says restarting R session. So everything's cleared out. See the environment is empty. The plots panel behind me is empty. But I haven't got my data. I haven't got anything here. You know, so now if I run, I try and make, you know, the head of this data set, it's going to say, error, can't find it. So how would I get all that data in? Well, I could run the entire chunk. But you see this little button that's right here. I hope maybe you can't see it too clearly in the video, but beside the little play green arrow that we've been clicking on for various code chunks, there's a button that looks almost like an eject key. What that does is it runs all previous chunks up to that point. So it'll run everything from above through the document. Boom, 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 boom. You see it's running all my stuff, loading, dseek2, going off and doing all our calculations that we've done thus far, and running them all. Okay, and I'm going to click try for this one. So it's doing the dseek analysis here, and then run it again. So it's sometimes if you're getting tired of waiting for this thing to appear, you've broken it. It was my fault that the thing ended up doing it multiple times here. So I quit it and then run it all again. And what we're going to do here is actually we'll save our script and we'll knit it to a PDF document. And that'll be us for today. Actually, that's our kind of session to end with this volcano plot. You can do the pathway analysis section. That will be optional. It's a nice section to end with, and we'll see how your gene sets, or how our difference expressed genes are enriched for different. Let's go back to the web page for a second. How they're enriched in certain pathways, and you can do that with KEGG or with Go. That's this section here. So we're section 7. Okay, so we install path view. These are the first few caffeine metabolism gene sets. There's other gene sets, and we map them versus the log to the fold change, and we can find out graft versus host disease, type 1 diabetes, asthma, upregulated, downregulated. Then you can go find these files or make little pin files in your project directory, and you can include those in your report. So what I'll do, I realize I'm 10 minutes over time here in our 2-hour session, and we might have a 3-hour time window here. I'm gonna call it quits here for now for the formal required part. The last little bit, I'll just finish and do this pathway analysis and show you how to include a figure in your document if you're so motivated to do that. But the formal part that you have to upload to Gradescope, I think once you have your volcano plot and you've answered those questions, those first 10 questions in your workbook, we're happy, we're good to go. So I'm going to go and so here we are back in, let's go back to our studio, see that we've check if we've fixed our, looks like we've got everything we need here. Let's run here to here, so I'm able to get ahead of it. Yep, got this. Let's see if we can make our basic volcano plot again. Yep, happy days. An enhanced volcano plot without breaking it. There we go. So we can see there's, if you're able to zoom in there, that's that crisp LD2 gene that the original authors focused in. It's just one of these genes, right? There's a lot of others that we can dig into and examine. And in the workbook, we show you how to dig into individual genes and also to look at these genes as a whole, this red subset here that are both high log2 fold changes and low p-values, to annotate those into the pathways to put some actual biology context on this subset. I'll finish off and just do that pathway analysis here in my workbook. If we've run out of energy for this, I completely understand, but let's finish off with pathway analysis. So the idea here is we're going to use, again, some bioconductor packages. We're going to use the gauge package in this case to do this analysis. And the gauge package is one of a whole handful of them to do this. And then there's another package that we'll use called PathView, which will actually make the little figures. If you haven't got these, you need to do the balance C manager install. Let me put this code in. So we'll use pathway analysis. It's also called aka gene set enrichment. The basic idea is we're looking, does your differentially expressed genes, are they enriched in certain, and I can try and spell correctly sometimes, certain pathways, non-annotated pathways like you'd find in your textbooks for cell biology and your biochemistry textbooks. So if you didn't have these, you could install them with the BIOC manager. I'm not going to include that in my Markdown document, obviously, but that's PathView and Gauge is the main package, and then the Gauge data. We're going to load those up. Library Gauge, and library Gauge data, and then finally the PathView one. So we'll call those up. So there'll be a little bit of printout here from PathView to tell you about it, what it was built on, that it calls KEGG. Database KEGG is, K stands for Kyoto. This is a database of annotated pathways, mostly related to metabolism, signaling, but also like cell biology pathways, developmental pathways as well. And we'll maybe just use KEGG for now. So we're going to look at the KEGG data set first, and we'll look at just maybe the first two pathways from this data set just to see what they look like. So this data is it's here. Let's look at the head of KEGG sets. Humans, there's other model organisms here. We'll just look at the first two pathways. The first pathway in this KEGG data set is for caffeine metabolism. These are the enzymes involved in caffeine metabolism according to KEGG. There's another pathway here, and other ones. We want to see which of these, remember these are entrées IDs. We want to see if these entrées IDs are a subset of significantly differently expressed genes that we've got. So we're going to pass Gauge a little vector of our log2 fold changes with those proper gene names, these entrées IDs. So we're going to need an input vector of, we'll use log2 fold change here, but you could also use, if this was other data for proteomics, this could be spectral counts or any numeric quantifier of how important these things are, with named entrées ID names. Because that's, we need the entrées format names because KEGG, as you just saw, uses entrées. So we're going to take that. So we're going to make a little vector called fold changes. And we're going to make sure that the name is, so we'll do fold changes. And we'll just give that, let's work with this one, log2 fold changes, and then we're going to set the names of fold changes fold changes. This thing we just did. And this is going to be equal to the res dollar ultras. If we have it. So that will be, if we look at the head of this, fold changes. This will just be a little vector with, so the second gene didn't have any fold change, but it will be it doesn't have the names on it. Let's check. Oh, I spelled it. There's the Yay, okay. So now these are the names of that vector, so I didn't see them before, so I knew there was a problem, and then the gauge function wouldn't work. It would shout at us or just give us an error message. Now I can pass this to the gauge function, and I'm going to save the results. So here, I'm just going to run gauge. I'm going to give it this vector of fold changes, and then the actual set, which is g set. This is going to be the keg data set, the keg sets.hs. So keg sets.hs, and we'll save this as keg res or something. So when I run this, it goes off, runs the pathway analysis. If I looked at the attributes of this result, it'll tell me that there's it gives me results as greater than, less than, so the subset that are in the greater than or the less than fold change set. I'm going to look at let's look at the less than set, and I'll look at the first three results. So let's do keg res dollar less. We could look at them all, or we could just do head. And we'll look at the top. Let's look at the top four sets or something like that. Top four results. Please include asthma. Okay, this one, for example, sounds reasonable, right? The asthma pathway is affected. So I'm going to take this identifier for the asthma pathway. This is the keg identifier for asthma. And then I'm going to use the pathway function to go and get the pathway for this gene set. So I'm going to do pathview, and then this function, if I remember how to use it, let's look at the help for a second. Pathview will take the data set, so that's my fold changes, and then it's going to take the pathway ID. I can give it pathway ID, and then that is going to be whichever pathway you want to dig into. I'm going to look at asthma. This is the identifier in the keg database for it, so I'm just going to paste it in here and look at my files menu, because what's going to happen when I run this is it's going to go off and put some files in my project, including this one, this pathview.ping. I open this up. If you just clicked on it here, it would open it up in your viewer. I'm going to insert that into my document here. I can do the kind of code to put this in, mark them as exclamation mark, the brackets like a pin in a link, and then which is this one. And if I do that, I'll have my figure in, and there's my results. It's a little small here, but I'm going to go and save this and knit my document and see how that turns out. It'll take a little while because it's doing all our work again. the knitter document, but it'll have my results figure in here, my pathway analysis figure. And you could go and explore other pathways that are upregulated and downregulated that come up in that analysis. So with that, I'll just wait for my PDF to appear, and then I'm going to quit while we're still breathing, just about. So it's running the seek analysis step. We're nearly halfway there in the last half, pretty straightforward. So while this is finishing, it's approaching the end of that 50% mark. I'm going to pop over here to look at the workbook. So this is the last section. I also have one bit where you can look at an individual gene, but I also have this session info that I include in the bottom of all these. This is to help with reproducibility. It tells me the version of R I used, what packages I had available when I was running all this work. And actually R is finished underneath, so I'll pop over to that in a second. But this you can include in your R Markdown document, and it will print out the same sort of thing in your report, because my web pages are actually just generated in the same way as these reports. So I'm going to have a look at my results. So I've finished. And what we see is our PDF. So here it is, lab week 9. It's got all the results. I'm going to scroll all the way down to the bottom. You can see my figures in there. Happy days. There's my figure. I have my volcano plot with the genes. You can see there's the crisp LD2 gene. There's many others in here that we could go and examine and try and figure out what the biology is telling us, what the data is telling us about the biology that's going on here, like some of these very interesting genes that we could use all the bioinformatics that we've learned about to find out more about and see how they feature in these different pathways. But for that, if we get our PDF report and we've answered those first 10 questions up to that end of that volcano plot section, we're good for this week. So I'm going to finish here this lab hands-on session. I'm just going to finish off by saying thank you very much for your attention, for sticking it out through this one. And what I did here in this session was I put quite a lot of material. It's quite a dense one, but it gets us right to the ability to have next week as more of a synthesis week where we can go through and review what we've learned so far, as well as have a bit of fun with some visitors from industry and get to pick their brain about different things that they think is important in bioinformatics in a different sector. You get enough opportunity to see people like me in academia. Let's hear from some folks that are working in industry and exciting bioinformatics and genomics project in a range of different companies, and how they enjoy their work and what they get up to and how they got into it and how they got their start and what kind of people they're looking for to employ. A lot of these folks that we'll meet, they actually run big teams and employ people and have a lot of students join them as interns and that. So with that, I'm going to finish here and encourage you to post questions on our Piazza forum, as always, as well as questions for our industry leaders. And we're going to finish finally right now. So see you next time. Bye now.